In replication, double helix-shaped DNA is uncoiled and the strands are replicated to form two identical DNAs. Double helix is broken down into single strands by helicase. Helicase is an enzyme used during replication to break the hydrogen bonds between the complementary base pairs into two separate strands. Complementary base pairing means that the nucleotides are arranged in relation to their opposite base. For example, thymine is paired up with adenine and guanine always pair up with cytosine in a DNA. In RNA, adenine pairs up with uracil. After helicase divides the DNA, a different type of enzyme called DNA polymerase comes in and attaches itself on a strand. It catalyzes two nucleotides to bond together so it produces a new strand. New strand is referred to the strand that is made by replicating old original strand, and the parent strand refers to pre-existing single strand that is being replicated. A term semi-conservative is used to describe replication because half of pre-existing DNA will always be conserved or remain the same. Transcription is a process where a section of DNA is replicated to produce RNA. A type of enzyme called RNA polymerase moves along the DNA and unzips it into two strands. As it unzips, it also bonds nucleotides that are floating freely around the DNA to its complementary base on the template DNA. When RNA polymerase reaches its terminator, it comes off and leaves. The strand that is being used as a template is called antisense strand. Opposite from antisense strand is sense strand, and it is same as RNA except uracil is replaced by thymine. After mRNA finishes replicating, it detaches itself and moves on to the next process, which is translation, where protein is made using codons on mRNA.
Translation is the second step of protein synthesis. After transcription, the mRNA exits the nucleus through the nuclear pore. The mRNA then enters through the A side of a ribosome. A tRNA with a specific set of anticodons and amino acids comes and binds with the codons of the mRNA. Codon is a set of three bases that determine the identity of the amino acid. A second tRNA brings a specific amino acid next to the first tRNA. And an enzyme connects the two amino acids using condensation, and the first tRNA will break off its amino acid and exit through the E site. The second will make more room for the next one, and so on, creating a polypeptide.